Welcome, dear students. Today in chapter 3 we need to finish our lesson today, or our chapter. And today inshallah we're going to learn more about different methods of contraceptive means to decrease the number of births. Let's start our episode today. What would happen if the ovum fertilized? This is a question, or that what we need to know it. I mean the formation of the embryo. Okay, so the corpus luteum, it will remain to secrete progesterone hormone, and after that the menstrual cycle stops till after birth. So the fertilization happen here, and the embryo, it starts to form. The corpus luteum degenerates during the 4th month, when the placenta grows and takes over its function. And placenta simply it's just a tube that connects between the mother and the fetus or embryo to transfer food and nutrients, and at the same time to take and to carry the carbon dioxide and other wastes from the fetus or from the embryo. Okay, this is a placenta, and we're going to discuss it later. And we have a give reason for here, very important question. The corpus luteum is a temporal gland. Temporal means what? Temporary. Okay, it doesn't take much more time. It's not permanent, but just a temporary gland. If the ovum isn't fertilized from 1 to 2 days, what happens? The corpus luteum degenerates. Okay, so the production of estrogen and progesterone will stop, and that causes degeneration of endometrial or endometrium of the uterus, and menstruation happen. And if the ovum is fertilized, the corpus luteum remains till the end of the 3rd month to produce what? Estrogen and the progesterone hormone. To do what? To increase the thickness of the endometrium. Okay, I mean on the other hand, on the other side, we have existence or presence of corpus luteum or absence of corpus luteum in case of fertilization, and in case of there is no fertilization, degenerates and it falls down. Fertilization. In our episode today, we need to discuss what the fertilization or the fusion of sperm and formation sperm with the ovum, and formation of what? Formation of the zygote. Okay, well, fertilization, it is the fusion of male gamete sperm with the female gamete ovum to form zygote, which is contains or carries the full of genetic material, which divide, forming the embryo. Fertilization also, it occurs in the anterior third part of the fallopian tube. Okay, and by the way, after the ovum is released in the 4th day from the beginning to bleeding, it will be ready for fertilization through 1 to 2 days. Okay, just take from 1 to 2 days. Sperm can stay alive about just not more than 3 days inside the female genital system. Only the head and neck of the sperm penetrate the ovum until it's separated away from the sperm. The number of sperms ranged between 300 to 500 million per ejaculation, each one ejaculated, and many of them lost during their journey to the ovum. The man is considered, if the number is less than 20 million, the man considered as a sterile. Okay, it cannot fertile the ovum. The sperm cannot fertile the ovum. Sperm secrete hyaluronic enzyme which dissolves a part of ovum that coat the ovum. And also, after fertilization, ovum surrounds itself with a coat that prevents the entrance of any other sperms. Okay, so we have the first coat, which is what? Hyaluronic acid. The second one after fertilization. Okay. What about the pregnancy and embryonic development? Okay. Well, after 24 hours of fertilization, zygote divides by mitotic division to produce two cells. They are called two plastomeric cells in the upper part of the fallopian tube. After that, they divide to produce 4 cells after another day. The cells divide to form mass of cells. This mass of cells is called what? Morula. Called what? Morula. The morula is pushed along the fallopian tube by the action of the cilia and also contraction of the muscles to reaching the uterus. What happens when they reach to the uterus? The morula is implanted, they are fixed in the folds of the endometrium or the internal line wall of the uterus, which becomes enriched with blood vessels and also glands for feeding and fixation the embryo at the end of the first week of the fertilization. During the first half of the pregnancy, the rate of division and growth is very fast, is rapid, where the organogenesis and differentiation happens. Organogenesis means formation of organs, okay? And differentiation happiness means the differentiation of the embryo into head, arms, and so on. The embryo becomes surrounded by 2 membranes, one of them called chorion and the other is called amnion. Embryonic membrane, they are very important. The first one, chorion. The chorion, the outer membrane and the outer surface of the chorion fuses and forms finger-like projections which are inserted in the endometrium or the internal wall of the uterus that form the placenta, in which the capillaries of both mother and embryo are connected together, okay? After that, the second wall, which is amnion. The amnion, the inner membrane. So we have 2 membranes, outer one chorion and the inner one is what? Amnion. The inner membrane, which is called amnion, the margins of the amnion join, forming what? The umbilical cord, about 70 centimeters in the length. The umbilical cord also extends from the intestine of the fetus to the placenta of the mother, or that connect between mother and fetus. The length of the umbilical cord increases to give much more freedom for the motility and movement of the embryo inside the uterus or cavity of the uterus. The amnion contains amniotic fluid that protect the embryo against shocks and wet and also dryness. The amnion fluid prevents the stacking of fingers and toes and also cleaning vagina before delivery. This is the importance of the amnion fluid. While the umbilical cord is rich in blood vessels that transfer food, oxygen, and water to the embryo and waste from fetus away to the mother. Okay? Well, we need her to know the function of the placenta. The placenta transfers oxygen, food from the mother's blood to the embryo and transfers excretory wastes from the fetus to the mother, kind of exchange. After that, the production of progesterone that preserves the endometrium. I mean, it keeps the endometrium healthy and strong. The progesterone stimulates and activates the maternal mammary glands to develop gradually and to be ready for lactation and production of the milk. And it helps also in stopping of ovulation or formation of ovum inside the ovary. We need here to note something. Drugs, harmful substances such as alcohol, nicotine, and viruses that pass from the mother's blood to the embryo's blood through the placenta that may cause a serious deformities, or the deformation of the embryo, and diseases may happen to the embryo. The embryonic development, we can divide it in the mother uterus into what? 3 stages, each one 3 months, okay, according to the 9 months. The first stage, we call it first 3 months, and they are what happens inside the first stage. The nervous system and the heart start their development in the first month, and after that the differentiation of hands and eyes. Differentiations mean what? They start to appear, okay, of hand and eye. All of that happened in the first month. After that, differentiation of the 2 sexes, testes after 6 weeks, ovaries after 12 weeks, okay? We have a difference between male and female, like 6 weeks for testes and 12 weeks for ovaries. At the end of this stage, the first stage, at the end of it, system of movement like the skeletal system and response to stimuli become established. They start. This stage characterized with rapid mitotic division. For what? For differentiation, as we said. And after that, the second stage, 4th, 5th, and 6th month, the ossification and support of the skeletal system. Ossification is mean what? It's mean formation of calcium, or deposition of calcium inside the bones, okay? Strength of the bone. This is ossification and support of the skeletal system. Complete development of the heart. So the first stage, the heart, it just starts to develop, while the second stage, complete development of the heart. The heartbeats can be heard. The doctor can detect the position of the fetus in the uterus to modify its position if it was a wrong position, if it is abnormal or something wrong inside it. The sense organs are completed and size increases. The third stages, or the last 3 months, 7th, 8th, and 9th one. What happened by the end of this stage? Growth rate slows down and development of the internal organs is completed, and after that brain development is completed. So the baby or the fetus ready to be delivered. In the 9th month, focus here, the placenta dissociates. It disintegrates, or it doesn't need it anymore. Dissociates gradually. So the progesterone hormone decreases, and attachment between the fetus and the uterus becomes loosened of the attachment between what? Between the fetus and the uterus. Labor begins with a series of contraction in the uterine wall under control of hormone produced from the posterior lobe of the pituitary gland. Labor, it means the signs of starting delivery. Okay, this is a labor. It's a group of contraction happen to the female before delivery. well, begins with a series of contraction with the uterine wall, the uterus, and all of that happen under the control of the hormone, okay, or effect of the pituitary gland. Fetus is expelled to the birth and ducked into the environment. The placenta separates and move outside. We don't need it anymore. The umbilical cord is cut and tied, or we need to tighten it or to fix it tightly. The baby starts his life with the crying to stimulate the air sacs and respiratory system to be ready, open, and to use. The pituitary gland secretes prolactin hormone that stimulates the production of milk to feed the newborn. With a valuable nutritive and emotional supply protecting himself from many somatic and psychic diseases in his future. Means of contraceptive pill. Well, what does it mean, means of contraceptive? How can we decrease or to lessen the number of birth in our society? Well, so means of contraceptive, we have a different means here, different methods. Number one, several contraceptive methods are used in the birth control. Like what? Like pills. So we have contraceptive pills. Contraceptive pills, they contain a combination of synthetic estrogen and progesterone. When the women start using them after the menstrual cycle and continue for 3 weeks, these pills prevent pregnancy by inhibiting ovulation. Okay, just it gives a kind of imagination for the ovary by presence of different hormones inside the body. Okay, it's just a kind of synthetic progesterone and estrogen hormone. Also, we have the intrauterine device or the coil, one of the most common methods, which is intrauterine device or the coil that inserted inside the uterine wall. It is inserted into the lumen of the uterus, I mean the inside the wall of the uterus, and it prevents the fertilized ovum to be implanted in the uterus. So the first one, which is contraceptive pills, it prevents the ovulation, while here it prevents the fertilized ovum to attach to the uterus. Okay. The last one is a condom, and it prevents the sperms from entering the vagina. We have also a different types like surgical sterilization for male and female. Okay, it involves the ligation or tying of the two fallopian tubes in the woman and cutting them, and sometimes we do need to cut them. And so the fertilization doesn't occur for the ova that produced from the ovary. One in the main, each vas difference is tied and separated from the other structures and is cut, so no sperms will come out. There is no sperms can ferment and or can be expelled outside the testes by tying what the vast difference in male and tying and cutting the fallopian tubes in the female. We have also multiple births. What does it mean, multiple births here? Well, usually one baby is born in each birth, and sometimes multiple births take place, reaching 6 babies in the same time. Yes, that happens sometimes. The most common are the twins. The international percent of twins is once in about 86 births. The multiple births are rare. Okay, so there are two types of twins. How many types of twins that we have? We have two types, okay? The two mature ova are separated. The first type, if the two mature ova are separated, the two ova from two ovaries at the same time, okay? From one or both ovaries at the same time, okay? The two ova are fertilized with two sperms. So we have here what? Two different ova, okay? And they are also fertilized with two sperms. Each one will develop to an independent embryo, okay? We have two different embryos. The two embryos differ genetically. There's no any similarities between them because they are formed by two different ova. Each embryo has its own embryonic sac and separated by its own placenta and its own feeding from the mother and so on. So each embryo has its own embryonic sac and separate placenta in the uterus. They never exceeded brothers of the same ages, okay? So they are separated. The other type, which is identical or monozygotic, we call it monozygotic what? Twins. This type of twins results from the fertilization of one ovum by a single sperm. We have just one ovum and just one sperm. The developing cell separates into two masses of cells, into two morula, and actually the reason of separating of them till now it's unknown. Each of which develops independently. They form two embryos, identical genetically, and share one embryonic sac and mostly one placenta. So they are identical twins. They have the same genetic characters, but not in everything, okay? These twins may be born partially attached to each other in some cases. Okay, we have here two different types, the identical twins and the other twins. They are what? Separated from each other, okay? We have here on the left side of the screen, we have two ova. They are maybe from the same ovary, or maybe what? From two different or from the same ovary, two ovaries, okay? They are fertilized by two different sperms. While the right side of the screen, we have just one ovum and fertilized by just one sperm, and it gives us what? Two masses are what they are developed. A case known as Siamese twins, which can be surgically separated in most cases, and sometimes it will be very hard. So we have here the test tube babies, okay, the last piece of information in our lesson. A mature ovum is obtained from a wife's ovary. Again, we bring what? A healthy and strong and mature ovum from the wife, okay? And being fertilized externally, okay, from the husband's sperms inside test tube. So we brought the ovum and the sperm outside the body, okay, and we keep them in safe place inside the tube, in a test tube, by certain nutritive medium to reaching what? To the morula, okay, to reaching what? To the morula, and after that it re-implanted in the wife's uterus to complete its embryonic development till birth. And this is called what? The test tube baby. And we have here what? The test tube or re-inclination, which is test tube babies. We have the health and the strong ovum or egg from the female, and the other side we have the semen that contains the sperms of the husband, and by fertilize them or being fertilized externally inside the nutritive material, they are form what? Fertilized ovum, which is zygote. After that, after formation of zygote or morula, they re-implanted again to the uterus wife. And this is the meaning of test tube babies. After that, we have something called renucleation. Let's discuss it. What does it mean, renucleation? Well, the experiments actually have been conducted on frogs and mice. Okay, the nuclei from amphibians or toads, embryonic cells, sure, at different stages of development were removed. Again, we bring what? We bring a cell and we removed the nucleus of this cell. From what? From toad or frog. Okay, and after that, we transplanted into unfertilized amphibian egg whose nuclei had been removed or destroyed by radiation. So we have what? We have here two cells. One, we brought one cell, and by removing this cell, and we re-implanted this cell in what? In removed ovum cell. Okay, they are maybe removed or destroyed by radiation or such a different way. The normal development was proceeded, giving rise to individuals identical in characters to the individual from which the cultured nuclei were taken. Yep, seriously, that happened in the labs, in the different labs, worldwide. This provides us what? That the early embryonic cell's nucleus is capable to direct the embryonic development in the manner similar to that of the zygote nucleus itself. And this is the meaning or the idea of what? Of renucleation. Finally, we have something called gamete banks, and it's a very nice and awesome idea. Well, the gamete banks are found in some European and American countries. These banks are used to store selected animal gametes, especially those of cattle and horses of the very rare strains or very desired strain, to keep them available for reproduction after that till the time we need them. Okay, the gametes are stored in frozen condition about -120 degrees Celsius Fahrenheit or up to 20 years. And they take much more time. We can freeze it more than that. After that period, that can be used in artificial fertilization, even after the death of the producer individuals, or if some rare animal species are liable to extinction, they may be dying out, they may be disappeared. Okay, we can what? We can use or take the gametes from gamete bank and do something like external fertilization as we studied, and to get the extinction species again. Okay. Also, certain people desire to store their gametes in such banks to ensure that kind of continuity of their generations even after their death with the several years. And that starts to be happen in the last years in America and some European countries. Also, some animals that researchers are carried out to control the sex of their newborn. And the question here is for you: could such a technique succeed in case of men? Yes or no? See you the next time and think deeply in the answer.